DNA replication is important because without DNA, cells would not contain the information to undergo mitosis. Without mitosis, humans could not grow or replace damaged.
So DNA replication is involved by separating two strands right here. So they are untwisted by topoisomerase. And then the helicase unzips the strands. And where they unzip is the replication fork. And the replication fork is where the hydrogen bonds are broken between the bases, and the single strand binding proteins hold the strands apart, so that way the hydrogen bonds do not reform.
Because DNA strands run on anti-parallel, there are two different processes involved in replicating each strand. The leading strand, which runs from 5' end to a 3' end, can replicate easy because the enzyme that adds nucleotides, DNA polymerase 3, can only add enzymes to the 3' end.
The lagging strand, which runs from the 3' end to the 5' end, is a lot more complicated. The reason why is because DNA polymerase 3 can only add to the 3' end, and since this end is the 5' end, we have to use a different method. So to start off, another enzyme called RNA primase adds RNA primers, as seen right here in the shaded nucleotides. And then the DNA polymerase 3 can add on to those. But what happens is these create these little fragments called Okazaki fragments. And so then we need to connect them together to make the strand whole. And to do that, DNA polymerase 1 will remove the RNA and replace it with DNA nucleotides, and then finally DNA ligase will combine them together. And then the replication of the DNA is complete.
Now, as you can see, the structure of DNA is a shape known as a double helix, and it looks like a twisted ladder.
Well, you see, DNA is composed of nucleotides, which are made up of a phosphate group, a pentose sugar, and a nitrogenous base. So as you can see over here. And then the backbone of the DNA is composed of a phosphate group and the sugar, which is over here.
The phosphate and the sugars are bonded together through covalent bonds called phosphodiester bonds. So these bonds are right here. DNA gets its twisted shape through the hydrogen bonds formed between the nitrogen bases right here. As shown.
And there are 4 kinds of DNA nitrogenous bases: adenine, guanine, cytosine, and thymine. And there are 2 categories of bases: purines, which have 2 rings, that's guanine and adenine, and pyridamines, which only have 1 ring, and that's cytosine and thymine.